So in this lecture, we're going to focus on the viral diseases of the nervous system. And recall that viruses, because they are so small, they are ultra microscopic, they're actually the more common organism that can cross the blood-brain barrier. So they're going to be more common than some of the bacterial and fungal infections from this chapter. So recall there were many species of bacteria that can cause meningitis, but in viral meningitis, most cases are caused by a group called the enteroviruses. And recall that the prefix entero means GI, so most of these are transmitted. One of the ways that they're transmitted is by fecal-oral route. So we'll talk about transmission here in a moment. But typical signs and symptoms are going to mimic those of bacterial meningitis. They're just not going to come on quite as quickly, and they'll usually be milder than what's seen in either bacterial or fungal meningitis. The virulence factors here are really just tied specifically to how viruses operate, right? They must get into our host cells, and the damage that they do to those meninges through just destruction of the host tissue cells is their primary virulence factor. So diagnostics, because their signs and symptoms are very characteristic of bacterial, it's just the absence of bacteria in that CSF sample, which is why viral meningitis is also sometimes referred to as aseptic meningitis. Another difference from bacterial meningitis is that viral meningitis can be seasonal, and we tend to see higher numbers in the summer and the fall when the weather is warmer. And it's also more common than either bacterial or fungal meningitis. It can be spread through respiratory droplets, but primarily we think of it as ingestion. So ingestion of either contaminated water from a poorly chlorinated pool, which is part of that seasonal factor, but it can also just be direct fecal oral route. So very oftentimes very young mothers get infected during the diapering process. So if the baby gets infected and then the mother gets infected from there. And treatment, because it's viral, that's going to be a theme throughout this whole lecture, is that there are no specific treatments available. We really just have to let it run its course. On average, it's a week to 10 days, but in some cases, viral meningitis is fairly mild and it can last for several months. Prevention, of course, primarily that fecal oral route can be prevented through good hand hygiene, obviously good cough hygiene to prevent the respiratory route, but otherwise it's usually a fairly mild infection. What most people just call polio is technically poliomyelitis, and this is caused by the poliovirus, which is also in the same family as the enteroviruses that we were just talking about. So signs and symptoms, it really depends on the individual and how far along the infection is. So four different conditions may be identified. So 90% of cases are completely asymptomatic. About 5% of cases can progress into what we call minor polio, where there's sort of vague symptoms, maybe fever and headache, maybe malaise, things like that. Another 2% might progress to what's called non-paralytic polio, which is where the meninges are involved and you might have muscle spasms or back pain. Paralytic polio is usually going to then further involve the spinal cord and control of skeletal muscles at various places in the body. So again, if the paralytic polio version, if the diaphragm is affected, then the individual could suffocate if they weren't put into the iron lungs. So we'll see what that looks like here. But about 80% of people that get paralytic polio might progress to a severe form that's very long-term, 30 to 40 years after they recover from the paralytic polio, can get what they call post-polio paralytic syndrome, where you have nerve damage and paralysis. So this is what they believe President FDR had. So of course he was confined to a wheelchair because of polio. So again, transmission, this is an enterovirus and is spread through contaminated water. And so this is a disease that is exclusive to human beings. And so where you have infected humans, it gets into their fecal waste. If it contaminates drinking water, then it just becomes a vicious cycle. And so in many places in the world where we've been trying to eradicate this, so for example, we eradicated smallpox. The World Health Organization has been working for decades to try to eradicate polio as well, through vaccination. And so one way, because it's not quite as obvious as smallpox when people are infected, what they do is they actually test sewage water for the presence of the virus, and then we know that it's whether it's present or not in a community. And so we were nearly to the point where the World Health Organization would declare this eradicated, but even as of let's see, 2015, they still had about 22 cases across places like Afghanistan and Pakistan and Nigeria. So we still have some places in the world where it's really difficult to eradicate because they're in very remote areas. In some cases, so for example in Nigeria, recall that when they did the ring vaccinations for smallpox, they would basically find a case and then they would go out and vaccinate everyone they came in contact with. Well, there were a lot of people that actually refused the vaccine and were forcibly given the vaccine back when they were doing that eradication. And so there's still sort of a stigma associated with the little white van that the World Health Organization, when they approach these remote areas, in many parts of the world, they run when they see that van. So it has been difficult, and there's still some propaganda in some parts of the world where they fear that the injection that they give you when they're really inoculating you against polio is actually that they're inoculating you with HIV, or that shot is going to sterilize you. And so it's a cultural misconception that has kept some of these areas from getting the vaccination fully engaged. So the iron lung was basically a pressurized tube that if your diaphragm was not working efficiently, that it made it very difficult to breathe. And so most people, this was very common for polio patients prior to 1935. And they might spend an average of 2 years getting daily treatments or just permanently residing in the iron lung. And some people it recovered if their nerves regrew and they were able to breathe adequately, they would remove them. But some people had permanent paralysis and spent the bulk of their life in the iron lung. So of course there's no treatment, no cure for this viral disease. You just have to let it run its course. And again, we just mentioned the 4 different conditions that might occur, and it really depended on your immune system. So we do have 2 very effective vaccines, but they each have their advantages and disadvantages. One of them is less stable without refrigeration, so that creates a problem if you're trying to get it to very remote areas of the world where you don't have refrigeration. And so that's why we have 2 different versions, and they're each used in their own particular areas, whichever one is the most efficient for that area. But that's the primary prevention mode is vaccination. And rabies. So this is a virus that has the scientific name the Lissa virus, and Lissa in Latin basically means frenzy. And so you can picture, a frothing at the mouth, an aggressive animal is in a frenzy, and that's how they named this virus. So this is a zoonotic disease where we get transmission from wild animals. And so pretty much most mammals are good hosts for this virus, but not all of them are affected the same, and not all of them can easily transmit it back to another host. And so depending on where you live, which species is of highest concern depends on where you live. So in this part of the country, so in the central U.S., skunks are the primary reservoirs. And so if you see a skunk walking around during the daytime, it's very likely that it is rabid and that it is misbehaving, right? So now, of course, if you see a baby skunk, it may have just been orphaned and it doesn't know any better. But if you see an adult skunk, that doesn't mean a fox or a a bat or other animals don't carry rabies. It's just the primary host in that part of the world. We also know that it was documented in 2004 that someone that was infected with rabies became an organ donor, and 4 recipients, so they transplanted lung, liver, and two kidneys to 2 different people, and so all 4 recipients ended up getting rabies from those organs. So we know that's a possibility as well. So the first signs and symptoms are going to be pain and itching at the bite site. Eventually, once the virus finds its way to the brain, that's when you're going to start seeing the neurological manifestations. So think aggression and frothing at the mouth. And so if the nerves that, again, affect either the diaphragm or if the brain stem is affected, then those involuntary actions can be fatal. So the virus will, wherever the bite is, the virus gets introduced by the saliva into the muscle cells, and then it will replicate in the muscle, and it will then jump across that synaptic cleft and travel down the motor nerve toward the brain. And so it really depends on how far away the bite site is from the brain, how quickly this disease will manifest. And so because it has to travel back through that peripheral nervous system, if you're bitten in the face, the signs and symptoms are going to come on much quicker than if you're bitten on the foot or the leg, because it does actually have to travel back to the brain. So the highest susceptibility, anyone in contact with wild animals, of course, animal handlers. The incubation period, again, varies widely depending on the distance that the virus has to travel. And so because there is a fairly slow, they initially named rabies a slow virus, because it does not only have to replicate, so it takes a while for it to replicate in the muscle cells, and then it takes a while to migrate toward the brain. And so in that period of time that this virus is starting to replicate, you can actually develop immunity to the infection if you get vaccinated right away. And so this is one of those diseases that we don't vaccinate everybody as a preventative. We vaccinate the animals to remove the highest risk exposure. And so diagnosis really depends on just knowing that you've been exposed to the virus. And so rabid animals, of course, and confirming the disease requires a slice of the cerebellum. And so when they take a slice of the cerebellum, they can find these really distinctive, what they call negri bodies, or these little black inclusions in those neurons of the cerebellum. So obviously it's post-mortem that we can actually confirm rabies. So, if it's a rabid animal, bringing the animal in for diagnostics is better than trying to diagnose it directly in the human, because it can only be done post-mortem. There are some antibody tests now that they can do on those tissues to confirm as well. So treatment, again, this is not something that we regularly vaccinate everyone against, but if you have an exposure, there's a process, so what we call post-exposure prophylaxis. And so it's going to include 2 types of injections. You're going to get the immunoglobulin, or the antibodies against the virus, and in the opposite arm, you'll get vaccinated. And so because, again, the incubation period for this virus is so long that if you get vaccinated even after an exposure, you still have time to develop an active immune response. Now, you get these 2 injections, but you're supposed to get them in opposite arms, because remember what antibodies are designed to do. They're designed to bind antigens, and remember what vaccination is. Vaccination is introducing the antigen into your body. So you're basically injecting the thing that those antibodies are designed to fit. So if you were to put them both in the same arm, the antibodies would just neutralize the vaccine, and you probably wouldn't trigger the immune response, and you wouldn't be able to lock up any loose virus that those antibodies were going out to find. And so people tend to misconstrue the idea of getting vaccinated in case you ever get bitten again. No, the vaccination that you're getting right after an exposure is protecting you from that exposure, because the virus is so slow that you'll actually be able to hopefully protect yourself from that initial exposure. So prevention, because the rabies vaccine, by the way, is not very user-friendly. There are multiple shots. They're very painful. Why multiple shots becomes a problem is because some people can develop an allergy to the other components of the vaccine. So this is one reason why we don't give, some of the vaccines that we have to give routine shots for, they always ask, have you ever had a reaction to this shot? Because sometimes it's the solvents and the other stuff in the vaccine that your body develops an allergic reaction to and can cause anaphylaxis. So it's better to just remove the link between us and the wild animals that are the natural reservoirs if we vaccinate our domestic animals. So dogs and cats are going to be removing that link. And then there's arboviral encephalitis. You probably know this more commonly as West Nile virus. And so it's part of a big family of encephalitis infections that are caused by what we call the arboviruses. And they got named that because they are arthropod-borne, arthropods as in mosquitoes. So this is a large family of viruses that can transmit or cause what we call encephalitis that's arboviral. So blood-sucking arthropods, which is in this case mosquitoes, can transfer or transmit these viruses. And so all arboviral infections are zoonotic diseases that we just happen to be susceptible to. So they are much more common in animals than in humans. And when they do affect humans, usually you have very mild cold-like symptoms, maybe weakness, fever, mental confusion. But because viruses are so small, they can cross the blood-brain barrier and cause encephalitis and infect the actual brain tissue. And so symptoms are going to be similar to meningitis, but they usually refer to these as neuroinvasive diseases because they are actually getting into the brain itself. So again, the natural hosts include birds, horses, rodents, cats, dogs, chimpanzees, and even alligators, although researchers still don't understand that one because their skin is so thick, it doesn't seem that mosquitoes should be able to penetrate their skin. But humans just are a susceptible host, and so when we happen to come by those mosquitoes that have been infected, then we can get infected as well. So here are some of the names of those arboviral diseases. So most of them, if you notice, have the name equine in the name because they are primarily a disease of horses. And so we have Eastern equine, Western equine, Venezuelan equine, and then St. Louis encephalitis and West Nile encephalitis. That's the one that most people are probably familiar with in this part of the country. And so again, it's really, these are zoonotic diseases that we happen to be susceptible to. So here's sort of over the last 22 years or so, the range of where those cases of West Nile virus have been documented, including here in Arkansas. So when you see the disease in this format, you can see that, we initially discovered it in about 2002, I believe, was the first case in Arkansas, as it was across the country. But there's also a seasonality. So of course you're going to see more cases in the warmer months of the year when you have mosquito activity is highest. So diagnosis can sometimes be tough, and it's usually a fairly mild and almost asymptomatic infection, but it is highest risk for very young and very old patients and immunocompromised. So if you see this suite of symptoms, then it might be of concern, and so they can test for antibodies against those different serovars of arboviruses in this cerebral spinal fluid if they suspect West Nile. So again, the rash that's formed is usually very mild, and it's on the trunk, and sometimes is only some patients get the rash. So sometimes it's difficult to decide whether this is what needs to be tested or not. So part of it is looking at the seasonality, looking at is it a high risk person, whether it gets diagnosed or not. So of course, again, being a viral infection, there's no antivirals, there's no cure, there's just supportive treatment depending on the symptoms. So prevention, just like any other vector-borne disease, avoiding contact with mosquitoes is the key to preventing the infection. So if you're camping, using netting or insect repellents, things like that, around your home, getting rid of things like old tires and planters, anything that holds even a little bit of water can serve as a breeding ground for mosquitoes. But the best way to avoid it is to take out the missing link, if you will, right? So we do vaccinate horses against those strains that they can carry. So if we vaccinate horses, we kind of remove that direct or the closest link where mosquitoes would then bring it from a horse to a human. And then there's Zika virus infection. So this another one. This is a fairly new, or at least recently recognized, right? They actually discovered the virus in 1947, but then they really didn't see many cases until about 2013. They saw a really high number of cases that popped up in French Polynesia, but even then it still wasn't very commonly seen until after the Olympics that were held in Brazil. Researchers there noticed a high number of cases of microcephaly in babies that were born, and so that really brought this to the forefront and helped us understand it a little bit better. So the pathogen is the Zika virus. It is transmitted by the Aedes aegypti mosquito, which, by the way, is kind of considered the cockroach of the mosquito world. It can live in virtually any condition. Even the tiniest amount of water can harbor those mosquitoes. And so the primary place that we worry about this disease is in the developing fetus. And so you can either transmit it during fetal development, and the highest risk for the most severe cases is if it's transmitted from the mother to the fetus in the first trimester, and that will can lead to what's called microcephaly, where the brain does not fully develop. But it can also be transmitted during the birth, through the birth canal, and those conditions would be very different than the microcephaly, but it can also affect the newborn. We do know that it's sexually transmitted, and there's still a lot of research going into how much the virus or how long the virus is viable in different body tissues. We know that it is most viable for the longest period of time in semen, which is why safe sex is something that they recommend for women of childbearing age so that they don't get infected early on. We do know that it can be transmitted in blood transfusions as well as organ transplants, and pretty much anyone in the asymptomatic phase can transmit it through these different various ways, and so it's problematic for that reason. So again, symptoms in children and adults are fairly mild. It's really the developing fetus that we worry about the most, but the symptoms usually, again, kind of vague. Things like fever and joint pain are sometimes hard to narrow down, but things like a rash and conjunctivitis can also be seen. So birth defects, again, if it's in the first trimester, has a very high risk for microcephaly. It can also affect the development of the nerves that regulate hearing and vision. It can impair growth altogether. There is an increased risk of Guillain-Barré syndrome, which can actually be caused by many things. Some other vaccines, some medicines can cause this, which basically you see a rapid onset of muscle weakness because of damage to the peripheral nerves. But Prevention, of course, involves, again, just avoiding the vector. So avoiding mosquitoes, using insect repellents, bed netting, things like that. Because this particular virus can be transmitted through sex, then condom use or abstinence can hopefully prevent that. There are no vaccines available yet, but there's a lot of ongoing research on this particular virus. And so in June of 2016, I believe it was, the Smithsonian actually published an article on research that they were doing where they genetically modified male mosquitoes and then released them into the wild to mate with the natural population of mosquitoes. And basically what was modified about them was that the larvae that are produced, if those genetically modified males were the ones that fertilized the eggs, is that any larvae that were produced would actually die before they reached the age to mate themselves. So there's not a lot of long-term data yet to see if that worked. A lot of ecologists, of course, look at that and say bad idea because you're introducing unnatural genes into the population. You don't know what other parts of the genome might have been altered. And you're also essentially taking out a natural part of the ecology. So as much as we don't like mosquitoes and know that they can transmit disease, they also serve as a major food source for things like bats, which are also going to help pollinate crops and things like that. So it's a vicious cycle when we try to step in and take out a piece of the whole puzzle. So again, as a viral disease, we don't have any specific treatments. There are no antivirals that we know are effective. All we can do is treat any symptoms. So we have had active virus transmission even within the U.S. We do have the vectors. That particular species of mosquito is actually an invasive species, but the cases that have been locally acquired in the U.S. were primarily in Texas and Florida, where those invasive mosquitoes are. Otherwise, all the cases are primarily travel acquired. But you can see why Brazil, where the Olympics were in Rio, is a high-risk area. And again, so most of the cases, the highest number of cases that were documented in the U.S. were between 2016 and 2017, and the most recent years where they've looked at those cases, we're down to less than 35 a year kind of thing, just based on awareness and prevention of mosquito bites.